Biofilms challenges and opportunities. Dr. Nigel Ternan, please.
Thank you very much for the kind introduction. I've got both hands full, so I'm not going to be able to do anything. I'm going to have to keep the microphone here. Okay, and breathe. Why am I so nervous? I don't know. Okay, I'd just like to say thank you to Lida for inviting me to come and talk today about some of the biofilm analyses that we've done in our laboratory. And we'll consider some of the challenges and opportunities of doing biofilm analysis type work in our lab. And I'll introduce you to some of the methods that we use and show you some of the outputs from that. And really what we're trying to do is generate some quantitative data from that. So I've got a laser and I've got a slide advancement. So just a little bit of background. Yeah, people have seen me around the place, and myself and Bill and James as well. So our context, if you take microbiology at Ulster, and this is not every microbiologist at Ulster, this is just a list of the three of us that are involved with SafeWater. So James Dooley, myself, and Bill, who's been upgraded to postdoctoral research associate today as opposed to any other title. And we're working on SafeWater with you, as you know. So what we do in our microbiology research group is we try to combine molecular biology approaches, sort of state-of-the-art genomics, proteomics, transcriptomics, and qPCR and RNA sequencing with more traditional bacteriological methods that have been used forever, like plate culture, and we also combine that with light and electron microscopy as well. We have a number of cross-cutting research themes with other niche groups, and I'm going to show you what niche is on the next slide. And we obviously have collaborations with external organizations, both nationally and internationally as well. And having discussed this with James and Bill, I think our primary expertise really is in the recovery of bacteria from different types of environments. And we also have a pretty good handle on working with difficult to culture microorganisms. So obligately anaerobic bacteria such as Clostridia, microaerophiles including Campylobacter and Helicobacter that cause problems in the food chain, and indeed organisms that we still actually can't grow in the laboratory to any appreciable extent, such as Cryptosporidium. So that's really our unique selling point, if you like. We can work with these difficult organisms. We have a lot of expertise in dealing with them, and we have the facilities. We're very lucky to have the incubators and the bits and pieces that we need in order to be able to do this kind of work. Within our group, current research themes, and these are always changing, so I chose some themes almost not quite at random, but they're fluid. So we want to understand how environments affect the transfer of antibiotic resistance determinants between bacteria. We want to understand as well how gastrointestinal tract bacteria in your gut maybe colonize and develop, maybe form biofilms. And in the case of certain pathogens, the biofilms that they form maybe lead to persistence and reoccurrence of disease. And we're also interested in novel methods to reduce microbial contamination, either in waters, obviously, which is why we're all here, but also in sensitive environments and on surfaces and various kinds of liquids. And we've done this kind of work with a diversity of industrial partners, short and long-term projects, direct consultancy, and obviously proper research-funded projects as well. So that's just a little bit about the microbiology at Ulster. There's at least 4 other colleagues who work on things like rhamnolipids, surfactants, clinical microbiology, salmonella, solid substrate fermentation, and things like that as well. So it's not just the 3 musketeers here. So I mentioned niche. Niche is the research group that we are part of. It's the Nutrition Innovation Centre for Food and Health, and it was established on the Coleraine campus about 25 or maybe 30 years ago by Professor Sean Strain, who stepped down as director only a couple of years ago. And our colleague Helene McNulty, who most of you will know if you've ever met her, she's now our director of niche, and there you go. That's who's the boss of us. So you can see here that niche comprises a variety of different disciplines. We have our teaching and our PhD students, obviously, but we also do research into vitamin D, gut health, aging, child development, agri-food, and in the bottom left-hand corner, we have microbiology as well. And we like to think that we make a valuable contribution to a lot of these other niche research areas, certainly in terms of primarily gut health and maybe immunology and stuff like that as well. Certainly aging and agri-food fall in there. So I meant to set a timer on my watch to see where I'm going, so might be a wee bit on schedule. So this afternoon, normally when we teach microbiology, we have 12 weeks to get people up to speed on everything they need to know about microbes. And I've talked for about 5 minutes at the minute, so this is actually less than 40 minutes. So it's going to be a whistle-stop tour of microbial growth and metabolism and getting on to biofilms. But primarily what I want to consider this afternoon is bacterial growth, talk a little bit about how we measure planktonic growth, where bacteria are swimming around, and also then how we measure biofilms, where they're stuck to a surface. And the main thing we want to try and get from our biofilm experiments is quantitative data. We're science and technologists. We're obsessed with numbers. So our social science colleagues might go, oh, there they go, with wanting numbers. But numbers is what we like to generate from our experiments. So we can say, yes, it is bigger or smaller. So as I said earlier on, I will look at consider some of the methods that we use in our lab to investigate biofilms and gene transfer. And then at the end, try and introduce a few future opportunities and the challenges involved with biofilm work, and maybe in the discussion that follows some new ideas might come from the group. So without further ado, bacteria are, I was going to say fairly simple. They're not, but they need food. They like to be nice and warm. They don't like it too acid. They like a lot of water. They need a bit of time to grow, and if they are aerobes, they like oxygen. So this acronym actually comes from the food industry, and I stole it because it makes things pretty clear. But when bacteria have these conditions, they'll grow quite happily. And growth proceeds from a sort of lag phase where nothing very much maybe appears to be happening, to a logarithmic phase of growth where the cells are growing very rapidly. And then the cells sort of run out of nutrients and they slow down and enter what we call a stationary phase, which is where nutrient starvation is happening and cell division is very low. And a lot of sort of students think, oh, well, nothing happens in stationary phase because the things that we measure don't change. Not true. A lot of interesting things go on in stationary phase, but we don't have time for that this afternoon, as I've said. So we can measure these parameters relatively easily using traditional microbiological methods of analysis. Oops, wrong button. Back we go. Find the laser. We can measure biomass directly and indirectly, and we can directly count bacterial numbers, just as Bill does when he's doing his water filtration stuff and counting colonies on plates. So these are what we call, this is for you, Patrick. This is a bugs in jars experiment. So it's a colloquialism that we seem to have developed over the last little while. So we grow bacteria in glass flasks, we keep them warm, we shake them around, and we can see that the optical density increases in both these growth curves. So we have a lag phase of about 20 hours, logarithmic growth up to 40, And then we have a very rapid stationary phase, and thereafter actually even a wee bit of cell death. So we can make these measurements with a spectrophotometer. We take a sample of culture, put it in a cuvette, pop it in the spectrophotometer, and we get a number, an optical density or attenuance. I was speaking to Helen last night and earlier today, and here's an example of biomass protein. So we can measure optical density, which gives us a fairly direct and immediate assessment of bacterial growth. And we can also look at protein in that culture or in cultures. Takes a little bit longer, but it's just another way of doing it. We're measuring the total amount of synthesized protein that the bacteria, or in this case a yeast, have actually produced, and it gives us, as you can see, the same basic shape of growth curve. So these kinds of experiments give us very rich, they can give us very rich temporal information. We can calculate cell growth rates, we can look at substrate and metabolite concentrations. This organism here releases phosphate, which it then uses during logarithmic growth. And this one here removes about 60% of the substrate that we put in that we actually measured in this experiment. So bugs in jars is our bread and butter. It's very easy to do, and you could take a reading every 20 minutes if you were so inclined, and get lots and lots of information. We can also do direct counting of bacterial numbers, and the schematic is here on the left-hand side. We take an Erlenmeyer flask, we take a sample out, we dilute it 1 in 10, and we dilute that sample over and over again. We then put some of the sample on an agar plate, and we incubate that probably overnight. And what we're aiming to see is somewhere between maybe 20 to 30 colonies all the way up to about 250 individual colonies on the agar plate. And we can do some simple sums and say, okay, we have 1.59 times 10 to the 5 bacteria per milliliter in this sample. And this is what, with the membrane filtration method that Bill and the other folks are using, this is what the sort of numbers that we are starting off with, perhaps, and ideally with SafeWater we see practically none at all. If we take it to its logical conclusion, we can do a growth curve and we can plate out, and you can see that both bacterial numbers and this optical density surrogate for biomass match quite nicely, and we can transform that into a kind of a, not quite a straight line, but it's not bad, a calibration curve. So if in this case we wanted to get some Clostridium difficile and we needed 2 times 10 to the 8 cells, we would probably have to grow the culture to an optical density of about 0.6 or 0.7, which is about mid-log phase. And this is the sort of calibration we use if we are extracting protein or RNA from cultures and we want to use a certain amount of cells to start off with. We can also do other things with the straight bacterial growth, bugs in jars. We can see here that we have 2 cultures, 2 different types of organism. We started off growing them at 37 degrees up to about 4 hours, and then we increased the temperature to 45 degrees. So you can see that the wild type continued to grow, whereas this DNA-K mutant, which lacks a key heat stress chaperone, it didn't like these conditions one little bit. The growth very quickly faltered, and in fact we've see a bit of cell death there as well. We can express our absorbance on a logarithmic scale. So there's a lot of things we can do with this way of actually measuring bacterial growth. And you can have log scales, you can have protein, biomass, direct counts, all sorts of things. Lots and lots of information. Find the right button again. So if we take this again to a sort of further extent, planktonic cultures are really the most widely used approach in microbiological research. They're simple and they're easy. You can do a lot of different experiments different ways. It's good fun to see how bacteria react to different conditions. We tend to collect cells maybe in mid-log phase because they're fast growing. It's a homogeneous culture. All the cells are at the same physiological state of being. And that means that if we take a sample we're getting a representative sample of what's going on. This is good if we want to do any kind of omics analyses, transcripts or proteomes, because we get a representative sample from the culture. And this becomes important when we move into looking at biofilms in a few moments. So we can make temporal measurements, we can change conditions, look at phenotypes, we can extract proteins, RNA, lipids, substrates and metabolites, and we can analyze these all relatively easily. So we grow organism at 37 degrees, do a mild temperature upshift, and we can see that for Clostridium difficile the graphs don't really diverge massively. This is obviously not the mutant that I showed on the previous slide. This colourful and complicated and very tiny figure is what we then did, and we did proteomic and transcriptomic analysis of this mild heat stress and published a few papers, and we can get some composite data from it that shows that for these very carefully selected genes at any rate, the direction and the extent of the change under heat stress is pretty much the same. So this is the sort of thing you can do with planktonic cultures and doing differential and quantitative analyses of different kinds. If we move on to biofilms, however, they're a very different beast. A biofilm, I suppose, can be defined as a bacterial population in which the cells adhere to each other or to surfaces and or interfaces with architectural complexity. So it's not just bacteria swimming around in a nice warm soup where they're lovely and happy. They're having to work for a living. They're having to attach to surfaces and maybe deal with low nutrient concentrations and all sorts of things. Biofilms are important. Clinically, many diseases are underpinned by biofilms. In cystic fibrosis, for example, there are biofilms in the lungs. With urinary tract infections and other things like that, you can get biofilms in there as well. Ophthalmic infections and, hip joint replacements, those kinds of things. If that titanium implant goes in, it only needs a few hundred bacteria, and you can actually get an infection that will lead to needing resection of that particular joint replacement. So stopping biofilms from forming is quite important. That said, biofilms also occur naturally. They occur on rocks in streams as part of healthy ecosystems, in association with other living organisms, microalgae and things like that, and slow sand filter material, obviously, which I think Helen is going to talk about a little bit later on. So a biofilm develops, there's a series of stages. We have these so-called surface naive cells. They've never seen a surface before. They stick to it. They're quite reversible. In stage 2, the cells stick to the surface irreversibly. So this is the start of the formation of the biofilm. And then we have various stages of maturation. And sort of towards the later stages of biofilm maturation, we actually see a release of the biofilm cells. So you can see if you had a biofilm, it grows, the cells get released. So even if the biofilm has been killed, it's released some cells, and that can perpetuate an infection or a fouling process in engineering systems or water treatment or what have you. So how do you measure a biofilm? It's not bugs in jars. This time it's bugs on microtiter plates, so these microtiter plates. And the way we do it in our laboratory, we will maybe have a pure culture on an agar plate. We take 3 colonies into 3 separate vials of broth. So we now have 3 biologically independent cultures. We grow those overnight, and we then inoculate them into fresh medium at about 1%. That inoculated media is transferred to a microtiter plate, 96 well, 12 well, 24 well, all can be used. We then allow that to grow for a period of time, 12, 24, 48 hours, that kind of thing. And we can measure the optical density in the wells to give us a planktonic reading. And then we drain away the bacterial culture, rinse the plate, stain with crystal violet, drain that out and rinse. And if you hold the plate up to the light at that point, you can see it's a little bit blue. So we want to measure the blue. We destain with acetic acid, and that solubilizes the dye that is bound to the biofilm that is stuck to the plastic. Then we can put it in a plate reader and get an optical density reading for the amount of absorbance. And the amount of absorbance that we get from the dye is going to be proportional to the amount of biofilm formed in that time on that plate. So that is whistle stop, and I hope that's clear to everybody. I mean, this assay was sort of first published by O'Toole and Coulter in 1998. But as you can see in the center here, we can look at biofilm for a couple of different types of organism and conditions. It probably wouldn't hurt if it was a little larger, but trust me, it's very pretty when you get up close. So that's how we measure biofilms, quite simply, and we can get some quantitative data as absorbance at 570 odd nanometers. But as I've said, they're very different beasts, and biofilms differ from planktonic cultures in sort of three main ways. As I've said, they are multicellular, and there is this 3D architectural complexity. So it's not just bacteria swimming around all doing their own thing. They're working together to form a structure. Metabolism is quite different. It's not homogeneous. If we take a mid-log phase planktonic culture, all the cells are probably doing the same thing. Within a biofilm, all the cells are not. And that makes measurements of gene expression and things like that a bit more challenging. And as you've seen, it's not just as straightforward to make measurements of biofilm. Planktonic culture, cuvette, you have a reading, well, depends how close the spectrophotometer is, but 30 seconds later. But with a biofilm, as you've seen on the previous slide, there's a series of processes, and those all take time. In addition, if you want to look at 24, 48, 72-hour biofilm, you need a plate for each of those time points because you sacrifice the plate when you make the measurement. 3D structure leads to this architecture. It's lots of different cellular phenotypes. The biofilm grows, gets thicker, maybe up to 160 microns or more. We have anoxia, no oxygen at the base of the biofilm. We have channels in the 3D structure that's a bit like a sponge, and it allows nutrients in and out. But despite that, antibiotics do not penetrate particularly well. And even if they do, within the biofilm there will be extracellular enzymes that maybe degrade antibiotics, and there'll be extracellular polysaccharide and bits of extracellular DNA and stuff like that as well. I think Helen's going to talk a little bit about the EPS. So within a biofilm, you actually have quite a large potential for the transfer of genes between bacteria, because if you have a mixed biofilm and one microorganism has a very handy antimicrobial resistance gene and another bacterium does not, they may well transfer that genetic material between each other. Most of the stuff that I'm going to show you this afternoon is single cell or single species biofilms. We haven't really looked at mixed microbial biofilms to the same extent. But the point is we can visualize biofilm structure microscopically. And I actually managed to say that word properly. So here we have, again, C. difficile. We have a wild type strain here. We have again, this DNA-K mutant that we constructed. And just using light microscopy with crystal violet staining on microscope cover slips, we can see that the wild type, some cells stick, but with the mutant we get a much thicker, denser coverage of cells on the glass surface. So this is very pretty, and it certainly shows us that the two bacteria types are quite different in terms of the way they form a biofilm and how quickly. But again, we haven't got any numbers yet. The right button. We can also look at this using scanning electron microscopy, which generates even cooler pictures. So if I can get this correct, on panels A and C, we have strongly adherent, this time Pseudomonas aeruginosa, short rod cells, and you can see that they're sticking. There's definitely they're producing something. It's not just cells, there's cells and a little bit of matrix or something like that here as well. And here we have, that's a lot of cells all stuck together. In panel B and D, however, we have a weakly adherent isolate. It doesn't form good biofilms, so we can see that on the surface you only get a few cells, and here we just see a monolayer of cells that have just about managed to hang on to the surface. So we can see that even between two different Pseudomonas isolates, the extent of biofilm formation can vary quite considerably. And still no numbers. Scientists and engineers, we want some numbers. So just to recap, metabolism in bacterial biofilms, as I've said, contains cells at every stage of physiological development. Increased resistance to antimicrobial agents and processes. We've slower growth. It can take up to 3 days to get maximum bacterial biomass on our surfaces. Phenotypically and metabolically, they're very different because planktonic cells will maybe reach mid-log phase in 6 to 10 hours. We also have a high level of regulatory complexity that leads to the formation of biofilms within bacteria. Just to scare everyone, we've seen Mike's TD diagrams. Well, if we were playing poker, I'd see his TD diagram and I'd raise him 3 quorum sensing systems. So we have a lot of regulatory complexity in the formation of biofilms, and I'm not going to go through this gene by gene because I might get run out of town. But I think it emphasizes the point that the regulation and the complexity of biofilm formation is pretty complex. So, as I've said, in terms of ease of measurement, we can't make direct measurements. We can do crystal violet, which is indirect, and it is the go-to method, no doubt about that. We can make direct observations and measurements, and we get lovely, pretty pictures, but we don't necessarily generate any numbers from these. As I mentioned earlier on, we must sacrifice our microtiter plate at 24 or 48 or 72 hours if we want to make an optical density measurement of crystal violet. So in our laboratory, you find piles of plates that have been grown and stained, and they're waiting for the destaining process to measure the optical density. And you also you hear students beating plates off the desk to get the liquid out. So it's good, that work is being done. So we need multiple replicates, large numbers of plates, so it's a little bit more complex than just simply bugs in jars. But all that said, we can actually derive quantitative data from our biofilm images. So what we use is a system called backlight. It's a bacterial live dead staining system, and I'm sure it's familiar to anyone that does a bit of microscopy in the audience. We have Cyto9, which green, and propydium iodide, which gets into the cells if the membranes are compromised, so we can say that those cells are dead or damaged. So live cells, lovely green. There are some dead cells in here, and I'll draw your attention to this chap here. And then we can make a composite image, and we can see that we're getting a little bit of yellow in this part here. So we can look at live cells, dead cells, and we can make a composite overlay and get a bit more information. Again, no numbers yet, but I'm coming on to that. Here we are. Yeah, this is a monster image. I put it into my PowerPoint and it increased the file size by about 25 megabytes. And the reason is because I imported it at full resolution because I wanted you to be able to see the important bits. We were looking at the effect of an organosilane surface treatment to try and reduce microbial attachment and biofilm formation. Now, if I can get this right, you can definitely see that there are red and green areas. So A and B, these two here, if there's an untreated surface, you get lots of biofilm. Hurrah. C and D, we have a treated surface at 24 hours, where there's maybe a few more red cells, and at 48 hours where it's almost a bit like the control. So we used the ImageJ software. My PhD student, not me. I just write the checks. So he extracted the green and the red pixels, and we can see that yes, in the untreated version, we have a lot of live cells, some dead cells, and at 24 hours on the treated surface with this organosilane, we have roughly equal numbers of live and dead cells. So the organosilane treatment is making some sort of difference. Unfortunately, at 48 hours, it seems to have lost its effect. But using this red, green, live, dead backlight staining, we can generate and extract some quantitative data from these nice images. So that's quite neat. If we get even more technological about it, for want of a better word, we can do confocal scanning laser microscopy. And this is some work that Elena Delagiani did a while ago. So we actually had a flow cell reactor, like a 3D little tiny flow cell. We allowed biofilm to grow in it, and we then analysed the biofilm, and some of these cells were stained with green fluorescent protein or red fluorescent protein. So we're not looking at a stain, it's actually intrinsic to the bacterium at this point. And the interesting thing here is that all these different Pseudomonas aeruginosa strains form a different looking biofilm under the same conditions. So these are quite traditional. You can see a 3D little hillock type of thing here. Something similar here. This is very different. And again, the other isolate there is different yet again. So we can use this sort of CLSM to build up three-dimensional information about the overall structure of the biofilm. And this is actually something I want to get back to doing, because it gives us a lot of information. Using the same technique, we're also able to show, as I said, most of this is single species biofilms, but we were able to show that a heavy biofilm former, the green cells, could actually entrap a non-biofilm forming Pseudomonas aeruginosa. And it didn't just stick to the biofilm, it stuck to the biofilm and then grew in biofilm mode of life. Even though left to its own devices, it didn't really form a biofilm. And if you think of the quorum sensing systems that are involved in this, I was rehearsing last night and it occurred to me that perhaps the red organism has a defective quorum sensing system, but in the presence of an organism in which the quorum sensing and biofilm development is definitely there, perhaps some of the signals get to the red bacterium and allow it to actually start to form a biofilm. Again, there's going to be a PhD student getting the bad news when I get home. Guess what you're doing next week? So we can also do some other quite neat stuff that we discovered a little while ago. If we grow biofilms and process them for scanning electron microscopy, this is back to C. difficile, which can be encouraged to form a fairly good biofilm. And we can measure crystal violet staining and show that things differ and build up and get bigger. But we discovered that during processing for SEM, the biofilms actually, they're being dehydrated and they crack. So we get these cracks in the biofilm like dried mud on a puddle. And as a result of that, we can actually take the stage in the SEM and we can tilt it to 40 degrees. And only our microscope guy is allowed to do this because he doesn't want anybody crashing the microscope. But having done that, we were able to actually, I can just about see it, so you probably can't, but trust me, it's there. We made some measurements of the thickness of the biofilm, And you can see the internal structure of the biofilm here as well. It's a bit like a pistizio or something like that, if you've ever been to Greece. It's a pasta dish. My Greek friends will kill me. But we're able to make these measurements, which are, again, a surrogate for biofilm thickness, because there is this squeezing like a sponge of the biofilm during processing. So it's not the natural thickness. It's been reduced. But this DNA K mutant here, again, on the right-hand side, every time we did this analysis and got a lot of numbers and statistically analyzed them, it forms a thicker biofilm than the wild-type strains, which corroborates the crystal violet staining and other means of analysis that we've used. So that was actually really neat. Can't wait to get writing the paper. I'll get that out there. So, where are we at? Yep. One of the more recent things that we've been doing, I think I'm okay for time, is looking at pheromone peptide-driven antimicrobial resistance transfer within enterococci. And one of my PhD students, Amy, who's also supervised by Bill and James, this is witchcraft to explain, but basically if the cell contains the plasmid, then the peptide will bind to the cell. And once that happens, the plasmid gets transferred to a cell that doesn't have the plasmid. I think that's as simple as I can make it. So we stain with wheat germ agglutinin, and we see cells, they're there, they're red. We have our pheromone peptide with carboxyfluorocene on it, and we can see that at time 0 and 2 minutes, we have a lot of green. And this indicates that the peptide is sticking to the cells that have the plasmid in them. It reduces a little bit at 60 minutes, but this is supported by the literature, which says that binding is pretty much immediate, builds up maybe a little bit over 30 minutes, and then decreases thereafter. And again, Amy has extracted the colored pixels from the image to produce a bar chart, and we can see lots of green, a bit more, and then it tails off. The grey bars are the controls, which didn't have the plasmid, and the peptide doesn't bind. It's a beautiful image, but it takes up half the wall, so I only showed a small part of that. So that's really quite neat, and I think we can use this to look at maybe gene transfer within biofilms and things like that as well. So, as I say, that was a whistle-stop Microbiology 101 on biofilms, and hopefully it hasn't been too chaotic. But if we consider the challenges in biofilm work, certainly in my laboratory, if we want to stain with coloured fluorescent moieties, then we need maybe antibodies to the antigens in which we are interested. It's easy to get antigen or antibodies to, for example, flagella and cell wall proteins and things like that. And that's fine. But the problem is those will always probably be there. And what I'm interested in is the things that actually change between different bacteria and different types of biofilm. So antibody availability is a little bit of a challenge, but we're making a little bit of progress. We're sort of limited to confocal, although at Ulster we have a STED microscope which uses ATO dyes, and well, you can add a couple of zeros to the consumables costs, but you do get super resolution on that. We're also interested in trying to look at gene expression within biofilms as well. Easy to do in planktonic culture. You take a sample, you extract the RNA, and you get it sequenced. And the microbiologists in the room are laughing because it's not just as simple as that. However, with a biofilm, we've got this tiny proportion of cells, and we have to get them off the surface, and then we have to extract the RNA without killing the RNA. And I think that's going to be a challenge, but we're going to try and find ways around it. And that's before we consider the heterogeneous nature of the biofilms. So the gene expression will be different throughout, so we may not get a representative answer if we do something like this. Again, we need about 10 to the 8 cells to allow this, and I don't know how many cells we can get out of a 96 well microtiter dish. So we might need to develop some new biofilm growth modalities. If we want to show gene transfer, with the enterococci, we simply allow the plasmid transfer to occur, and we select the transformants on antibiotic containing agar. Very simple, very straightforward. We can calculate transfer efficiencies. But if we're doing this in a biofilm, we've got to disrupt the biofilm to get individual cells that we can then plate out onto agar and see how many we've got. And that disruption may completely destroy our ability to count the transformed cells. So we might need to develop a better system for that. So, I mean, and I suppose those are the three main challenges that there are many more, but three to start with, I think, is enough to get on with. And in terms of opportunities, there are other ways of growing biofilms. We can use this so-called colony biofilm assay, which is very good for looking at antimicrobial resistance and susceptibility in biofilms. And there's various models that would allow us to grow bacterial biofilms in the presence of and interacting with eukaryotic cells. So we could use gut epithelial cells, put in some C. difficile and see what happens. And we have a couple of PhD students starting hopefully in September that will be beginning to look at that. So I'm excited about that. Other things like flux in water treatment processes and on filters. Well, a glass slide is easy. You grow, you take it out, make some measurements, it's all good. A sand filter, that's going to be a bit more difficult maybe to get maybe samples from and get them in a state where we can analyse them. But I think Helen's going to tell us how to do that in a little while. So that's, I suppose, the challenges and opportunities as I see them, at least as of today. Tomorrow might be different. And I suppose at this point, as I've already alluded to, I didn't really do very much of this at all. It's all been done by PhD students. So starting with Elena and working all the way up to the more recent additions to our laboratory who are just starting their PhDs. And obviously not forgetting Bill and James and Patrick Naughton, who's right there. And Barry O'Hagan is our microscopy guru. And it'll kill me for showing this because it's about 25 years out of date. He looks a lot different now. And there's Bader and Elna. And Megan has just joined us. And Shalish has grown the biggest pair of glasses I've ever seen since we saw him last. So these are the guys that did the work. And I thank them very much for their efforts. And to conclude, I'd like to just say thank you to GCRF for funding SafeWater and to our partners in South America and the United Kingdom for being a great bunch of people to work with. I'm delighted to be involved with SafeWater and I'm excited about it. And I still am, which I think is good. And I'd just like to, at this point, extend my thanks to Lida and the team at USP for organising this meeting. It's been absolutely superb, and the organisation is puts us to shame. So thank you very much, Lida. Thank you, everyone.
Thank you, Dr. Nigel. 5 minutes to questions, please. Any questions, any doubts?
Nigel, where can I find a biofilm in this room?
On your teeth.
So they're everywhere.
Yeah.
This is not something new or wacky or crazy.
Yeah.
Biofilms are the way that organisms live in the real
They
world.
prefer to live as biofilms. With the way we grow them in the laboratory as planktonic cultures is actually really nothing like they grow in nature. If you go to any environment, you will be able to find something slimy, even your own shower or bathroom, all sorts of places that you maybe don't want to tell people about in your home, but you'll be able to find biofilms because anywhere there's a surface and a little bit of nutrients and some moisture, bacteria will quite happily stick to it and grow.
And this is one of the complexities of working with water again, because you might kill the bugs that are in the water,
Yep.
but along all the insides of the pipes and on the taps, if there are biofilms living, they'll recontaminate the water again.
Yeah,
So
and I
it's a
mean,
very tricky beast or community of beasts to try to work with.
yeah, yeah. I mean, some of the stuff we saw yesterday with the blue barrels, septic tanks. I mean, okay, that's designed to generate a microbial community, but even the pipes there, they will over time become. There will be biofilm on them. Whether that's a positive thing or a negative thing, it depends on what the biofilm does. If you have water butts connected to your house, every now and again you're going to have to take that pipe off and go to get rid of the goop that's built up inside it, and maybe that's the way of getting enough biofilm to do RNA extraction.
You know the way we say chlorine is great, chlorine kills everything. What does chlorine solutions do for biofilms?
That's a very good question. We've looked at biofilms, and we've started to get into trying to work out how we disrupt biofilm formation or remove biofilms, and ibuprofen's good. Yeah, you didn't expect me to say that. But we did an experiment during the summer with a Nuffield school placement student, and they put some ibuprofen in on top of some biofilms, and it reduced the amount of biofilm, or increased it. I need to look at the data, but it made a difference. So yeah, I mean, chlorination is an obvious thing to look at, and you might imagine that it's gonna. If the chlorine kills the bacteria, then the bacteria can't really stick, so it might reduce it. But again, the acceptability of chlorine is going to be a problem.
Thank you,
Am I off the hook?
Nigel. Just a silly
Sure.
question. What's the lifetime of the biofilm? It depends of the microorganism?
I think
or
it's going to depend on the microbe. With Clostridium difficile, we see 24, 48, 72. What comes in between? And then there's 96 and 120. It goes up at maximum at about 72 hours and then decreases thereafter. So because we're interested in doing quick laboratory experiments, we grow them to 3 days and don't worry about the next days. In nature, you've less nutrients and lower temperature potentially. So, biofilm will build up over time in a sort of natural scenario, and the extent of biofilm formation is going to be limited by, I suppose, the amount of time and the amount of nutrients in that particular environment. If you get runoff, you get eutrophication, you maybe get a lot of biofilm and algae growth in a water body quite quickly. But under sort of normal processes, it's quite slow. We artificially accelerate growth in the laboratory because we lots of rich nutrients and complex media, just because we want the bugs to do it quickly for our convenience. But that's a good question.
Thank you.
Worth investigating.
Thank you.
Oh, I'm for it now.
No, first of all, congratulations, because that was extremely interesting.
Thank
I'm not in the area at all, and I think that reached the purpose, which is try to capture the attention across different disciplines. I was actually trying to write a report, and then I got suddenly I was paying attention on biofilms. So congratulations, that fits the purpose of the exercise. What are we doing to guarantee sustainability? For instance, you mentioned in the pipes that they will be growing as well. That's a very general question. Yeah. But after the prototypes are installed and we are no longer having maintenance during the time, how we guarantee that that is not an issue?
I think it probably will be an issue. And, that's just occurred to me right now, the same way last night an experiment occurred to me. If you have a surface and you have water and you have microbes, you're going to get a biofilm. And I suppose whether or not that biofilm becomes a problem, it'll need a little bit of time maybe to determine whether it actually is a problem. If you have a filter and you have bacteria that are filtered out of the water, and they're still on top of the filter. They're not just going to sit there. They're going to go, hey lads, let's grow. So you could see I think, possibility of contamination of filters with bacterial biofilm, and I think it's something that maybe has to be considered. And I'm looking like I've just thought about it there now, but it's only because you mentioned it. And the confused look is because I'm trying to come up with a solution right now and I can't. But yeah, I think that's going to be, it's an issue in any water treatment process, anything like that, you will get biofilms. We did a piece of consultancy about 20 years ago for a company that had this red stuff, they called it, on one of their industrial machines. It was a cutting machine, so there was cutting fluid and moisture and temperature, and it was a bacterial biofilm. And we identified that what was in it was not particularly harmful, and that they told the company that they'd be quite safe to just wash it away from time to time and it wouldn't do any harm. But yeah, I think with the prototypes, yeah, I think over time there will definitely be some biofilm develop. So that needs to be taken account of. And I don't know how, but I'll think about it.
You will find out into the end of the project, right?
Yeah.